अस्सलाम वालेकुम माय डियर स्टूडेंट्स। आज हम बात करने जा रहे हैं फीमेल गेमेटोफाइट डेवलपमेंट की। फीमेल गेमेटोफाइट डेवलपमेंट इज आल्सो नोन एज मेगागेमेटोजेनेसिस because in this process the large gamete, or you can say megagamete or female gamete, is formed because female gamete is large as compared to male gamete. That is why this process is also known as megagemetogenesis. Okay, so the development of female gametophyte or embryo sac from megaspore, this is known as megagemetogenesis or embryo sac formation, or you can say female gametophyte development. There are three types of embryo sacs in angiosperms based on the development. Depending upon whether 1, 2 or 4 megaspores are involved in the formation of embryo sacs, accordingly we have monosporic, bisporic and tetrasporic types of embryo sacs. The monosporic embryo sac, it develops from a single megaspore. That means out of the 4 megaspores, the 3 megaspores they degenerate, and the single megaspore it forms the embryo sac. Since monosporic type of embryo sacs, they are formed in 70% of the angiosperms. Okay, they are formed in 70% of the angiosperms. This is the most common type of embryo sac development. We will here just focus on monosporic embryo sac development. Okay, we will not talk about bisporic and tetrasporic embryo sacs. We will just focus on monosporic embryo sac development. So you know that monosporic embryo sacs, they result from just divisions in a single megaspore. That is, only one megaspore is involved in the formation of embryo sac. The final organization of the monosporic embryo sac is such that it contains 8 nuclei and 7 cells. Or you can say it is an 8 nucleate 7 cell structure. And this type of embryo sac, it is also known as polygonum type of embryo sac because it was first reported in Polygonum. So polygonum type of embryo sac, or you can say monosporic type of embryo sacs, this is the most common type of embryo sac found in 70% of angiosperms, and that is why we are here discussing this type of embryo sac only. Now, how this embryo sac formation takes place? First of all, you know we have a single functional megaspore. Actually we have 4, but these 3, the lower 3, they degenerate, and the upper one, it functions, it increases in size, it grows in size and undergoes first mitotic division, resulting in the formation of 2 nuclei. One is directed towards the chalazal end and the other one at the micropellar end. Then each of these 2 nuclei, they will divide again by mitosis, resulting in the formation of 4 nuclei, 2 located at the chalazal end and 2 at the micropellar end. Each of these 4 nuclei, they will undergo the third mitotic division. They will result in the formation of 8 cells, 4 at the micropellar end, 4 at the chalazal end. Now, one very important thing is, out of the 4 nuclei at the micropellar end, the 3 they form the cells which constitute the egg apparatus. 3 form the cells at the micropellar end, constituting the egg apparatus, containing 1 egg in the middle and 2 lateral synergids. The remaining 1 cell, it moves towards the remaining 1 nucleus, it moves towards the center of the embryo sac and constitutes one of the 2 polar nuclei. Yaad rakhna hai, it constitutes one of the 2 polar nuclei. Similarly, at the chalazal end, 3 nuclei, they form the antipodal cells, and the remaining 1 nucleus, it migrates towards the center of the embryo sac and forms the second polar nucleus. Okay, so 3 nuclei, they form the egg apparatus containing middle egg or oosphere and 2 lateral synergids. Similarly, at the chalazal end, we have 3 3 antipodal cells, and at the middle we have 2 secondary polar nuclei. These 2 secondary polar nuclei, they fuse with each other. They form a single cell. Okay, they form a single cell here, and this single cell contains 2 nuclei. It contains 2 nuclei. That is why we say that the final organization of the female embryo sac is, it is a 7-celled 8-nucleate structure. We have 7 cells, 3 here, 3 here, and 1 in the middle. And this middle cell contains 2 nuclei. Okay. So before fertilization, we have 7 nuclei. Sorry, we have 7 cells. We have 7 cells. Out of these 7 cells, the one in the middle, it contains 2 nuclei. It contains 2 nuclei. Now let's look at the details of the female embryo sac organization, how the cells are organized. Okay, first of all, we'll talk about the egg apparatus. The 3 cells located at the micropellar end, they constitute the egg apparatus. The centrally located cell of the egg apparatus is called egg cell or oosphere. The lateral 2 cells, they are called as synergids or help cells. These cells, they form the filiform apparatus. They form invaginations at their upper wall, and these invaginations, they are known to attract and guide the pollen tube. Now the polar nuclei, it is a large binucleate central cell. A large binucleate central cell. Generally, both the polar nuclei fuse before fertilization and form a single diploid nucleus called as secondary nucleus or definitive nucleus. Yaad rakhna. Generally, in most of the cases, it has been found that the 2 polar nuclei which are in the central cell, they fuse before fertilization. They fuse before fertilization and form a single diploid nucleus. And that single diploid nucleus is called the secondary nucleus or definitive nucleus. Now the antipodals, the 3 cells at the chelazal end, they are known as antipodal cells. They are the vegetative cells of the embryo sac. They are the vegetative cells, okay? They don't have any role in fertilization. These cells, they generally degenerate soon after fertilization. When fertilization takes place, when the female gamete fuses with the sperm or the male gamete, right after this event, the antipodal cells, they degenerate. Here you can see this is the final organization of the female embryo sac. This is the egg apparatus. It is constituted of a single oosphere in the middle and 2 lateral synergids. These lateral synergids have the filiform apparatus at their upper wall. These filiform apparatus, they guide the movement of pollen tube. They attract and guide the movement of pollen tube. Right in the middle, we have 2 nuclei which are known as secondary polar nuclei. These secondary polar nuclei, they are present in a single cell. Okay, they are present in a single cell, and just before fertilization, they are known to fuse with each other and form a single diploid nucleus, which is known as definitive nucleus. Okay, यहाँ पे secondary polar nuclei, they fuse and form a single diploid nucleus. And here at the chelazal end, we have 3 cells which are known as antipodal cells. They are the vegetative cells, and these vegetative cells, they degenerate once fertilization takes place. Okay, once the female gamete fuses with the male gamete. Okay, so this is the final organization of the female embryo sac. I hope you all understand this. See you soon with a new lesson. Till then, stay blessed, stay safe.